Greetings! We are going to start focusing on more about cells and cell processes in this series of videos. And we're going to focus first on the idea of molecular movement. Why focus on molecular movement if we're talking about cells? Cells are always having to deal with internal and external changes in their environment. One of the things that's always happening is molecules, the things that are surrounding cells and the things that are in cells, are always moving. Why do they move? Well, everything has some energy, and our measure for energy is temperature. So temperature does measure molecular motion or molecular energy. When it's cold, molecules are moving slower. When it's warm, molecules are moving faster. So molecules are always moving. They're always bouncing off one another, and they're moving around. And cells have to deal with this. If cells were to not have some way of regulating molecular movement, cells are going to quickly die because they have to control what comes in and what goes out. But first, we'll focus on osmosis and facilitated transport, after transport in other videos. But let's go ahead and consider what diffusion really is. Diffusion is the random movement of a solute from an area of higher concentration to an area with lower concentration within a solvent. That sounds really fancy. Diffusion is really molecules moving from higher to lower concentrations. So when we look at some terminology here, we see some words that may bother you, but they shouldn't. A solution is any mixture of liquids, gases, or solids that something can move in. If you want to think of it as water or think of it as air, that is a solution. Air is a solution. Your blood is a solution. So it's anything in which materials can move. A solute is a particle, is a molecule, is anything. You'll hear me refer to it as stuff. It is anything that can move in solution, and a solvent is usually the liquid part of that solution. And a concentration gradient refers to differences in the concentration of stuff. That is, in some parts of the solution you may have a lot of a particle, and other parts you'll have less of a particle. Diffusion says random movement, random collisions of molecules bouncing off of one another is going to eventually cause an equilibrium, an equal spread of those solute particles in solution. In other words, think about, we'll look at a slide. That might make it easier. So let's start here. We have here a solution. It is some water, and we put in a purple dye pellet. This purple dye pellet has a lot of purple dye molecules in one spot. I drop it into the water. Well, the water molecules are bouncing off the dye pellets. The dye pellet's bouncing off the water molecules. And eventually the dye begins to spread throughout the solution. It is not spreading because it wants to. These are dye particles. They don't want to do anything, but they're being randomly spread by these random collisions. This is diffusion, because all molecules, again, have some energy. Kinetic energy means they have some energy as they bounce around. So the result is the material, the solute, is spreading from higher to lower concentration. It's bouncing around. Eventually, I'm going to have an equilibrium where the purple particles here have spread throughout the solution. So at this point, then, are these dye molecules still moving? Your initial answer might be, well, no, they've stopped because they've spread about. Actually, they are still moving, but there's no more concentration difference. So we don't see any change in their movement. We don't see any change in their movement because they have equally spread, but they are still moving. They don't get there and stop. Molecules are always moving. In this case, they're spread out. So we've said these particles have reached an equilibrium. So, can diffusion occur through a cell membrane? We were talking about it bouncing around the outside, but can it go through a cell membrane? It depends on three circumstances. One is lipid-soluble molecules can diffuse directly through the plasma membrane if there is a gradient. Okay, so over here you see lipid-soluble molecules. Why does it have to be lipid-soluble? These purple balls here are phospholipids. And we have a rule in chemistry, or a principle in chemistry, is like dissolves like. So it is very possible that you can have some diffusion where these particles can go from higher to lower concentration and pass into or out of the cell membrane because they're lipid soluble. So the answer is yes, for some things, because they are lipid soluble, they may be able to pass through the cell membrane. What if they are non-lipid soluble? Well, if they're not lipid soluble, they're not going to mix or combine very easily with phospholipids of the cell membrane, so they're not going to pass through. And as we said in class, this gets to the idea that the cell membrane is selectively permeable. It will not let non-soluble, non-lipid soluble molecules pass through, so in this way they're going to kind of bounce back off. So even though there's a concentration gradient, I got a lot more of these here than in here, and random movement would say they're going to bounce into here some way. They're going to bounce in and then bounce right back out, bounce here, bounce right back out, because they can't pass through. So here's how the cell membrane practices some of its selective permeability. Another way they actually can pass through, is that specific non-lipid soluble molecules can pass through particular channels. So here's a membrane channel. These membrane channels are proteins, and this protein just so happens that it's going to let these particles pass through from higher concentration to lower concentration, and therefore allow them to pass into the cell membrane. So diffusion may still occur through a cell membrane through these particular channels if this protein channel lets these particles pass through. Now, if I have some different particles, let's say I've got some blue ones here, these blue ones, these are not going to be allowed to pass through because they're not going to have the right shape. And so they can't pass through there because the protein won't let them. Once again, the cell membrane is selectively permeable. We do have a term for this though. Since this protein is helping them move through here, we call this facilitated diffusion. Facilitated means to assist. So these proteins in the membrane may allow some diffusion to occur. This is facilitated diffusion. I just want to give you some examples as we wrap up this video. So does this really happen in the body? I mean, why do we care about diffusion? There are lots of good examples. I just listed the first 5 I thought of quickly. Gas exchange between cells and tissue fluids. When we breathe in and we breathe in oxygen, the oxygen we breathe in, and we'll do a real silly diagram here. Here's oxygen in part of your lung. Here's going to be a blood vessel coming by. It's going to have very little oxygen in here, and there's going to be very little CO2. Kind of hard to draw this, and a lot more CO2 in the blood. Why the difference? Because this blood has come from the tissues, and the tissues have given up CO2 and have used up most of the oxygen. Here in the lung, that's supposed to be part of the lung, you have this concentration difference. Oxygen is going to move from a higher concentration into the tissue fluid, and then eventually into the blood. CO2 will move into the tissue fluid, and then eventually into the lungs because of the concentration differences. So diffusion, high to low concentration, is going to be really critical here. Some other examples, we see this in the nervous system as acetylcholine, a neurotransmitter, is released from the end of a synapse. It's going to move across a gradient and then affect the other side, affect the next nervous, the dendrite part of the nerve on this side, as particles move from higher to lower concentration. Digestion, as we eat and break down through catabolism, have our particles in our lumen, in the space of our intestine, they will move into the villi in the bloodstream because you have lots of particles in the lumen and very few particles in the bloodstream. We'll talk about more about urine formation a little bit later, but I gave you one more example here with the placenta. This is showing you how the placenta works. The blood that's coming from the mother is going to be high in oxygen, high in nutrients, very low in wastes. When it gets near so when it enters the placenta, there's going to be gas exchange because the blood of the fetus is going to be low in nutrients because the fetus has used them up, high in CO2 because the fetal cells have produced it. So there will be an exchange from high to low of those particles of gases and nutrients, which is how the placenta works. So a very important process, diffusion. Okay, we'll move on to osmosis in the next video. Before I do, forgot about this, you may want to pause and answer these questions. I think you can answer these without help, but I'll pause here in a few seconds and then I'll give you the answer. So if you want to pause, we'll do it in 3, 2, 1. And we're back. If you didn't pause, go back and do it. But the answer is here, why would lipids diffuse through the cell membrane but not other substances cannot? Rather, that's because lipid-soluble things can dissolve in lipids, other particles cannot. Diffusion is a passive, not active process. That means it's true. It does not require any energy. Particles are naturally bouncing off each other. And neither the concentration gradient nor temperature would not affect the rate of diffusion. That's not worded very well. Concentration gradient and temperature both do affect the rate of diffusion. Sorry, that wasn't worded too well. That's it. Moving on to osmosis in the next video.